everyone, I welcome you all to my channel. Today we will talk about stereochemistry of carbohydrates. The functional group as well as the covalent bonds of a biomolecule are very important and they affect almost all the properties of a biomolecule. That is, this is a biomolecule, a carbohydrate, a glucose molecule. The covalent bonds in this molecule, that is, let's suppose this covalent bond. This covalent bond, its placement is very important and it affects the properties of this molecule as well as the functional group. That is, in case of glucose, we have aldehyde as a functional group. In case of fructose, we have ketone as the functional group. So these functional groups also affect the properties of a particular biomolecule. But apart from these two things, the three-dimensional arrangement of the subsequence is also very important. That is, this is the main chain of the carbon atom. The subsequence, that is HOH, how they are arranged in three-dimensional space, that is very important. Three-dimensional space arrangement means, as I have already explained, that a biomolecule in three-dimensional space has a structure, and which when we want to draw that structure in two-dimensional space, that is when we want to draw a molecule of glucose on a piece of paper, then that becomes a bit different because we try to depict that three-dimensional structure on two-dimensional space. So we follow some rules, but we cannot draw the exact thing. That is, suppose this is our glucose molecule. In three-dimensional space, it has a structure like this, then it has a circular configuration. So these things, when we want to draw one piece of paper, we won't be able to draw everything. We will just, whatever we do, we will be able to draw, or we will be able to represent this pen in this form only. So the three-dimensional arrangement of things means the constituent atoms is very important, and that affects the properties of that biomolecule a lot. As you all know, in living system, the biomolecules, they interact. That is, let's suppose this is a cell, this is a receptor which is protein in nature, is present on the plasma membrane of the cell, and let's suppose this glucose acts as the hormone which will bind with this receptor on this side. So this is the binding site for this hormone, and it will bind over here. This molecule will be able to bind with this at this position with this receptor only when it is complementary. That is, the shape of this molecule should fit within this space. Then only this molecule will be able to bind with this receptor. Let's suppose instead of D carbohydrate or D glucose, L glucose tries to bind with that space. It won't be able to bind because there is a difference between D and L. In D and L, the subsequents are arranged differently at carbon number 5. Carbon number 5 is the farthest chiral carbon, and in case of D-glucose, as you already know, in case of D-glucose, the H and OH of the farthest chiral carbon are similar to D-glyceraldehyde, that is, OH is on the right and H is on the left. But in L, OH is on the left and H is on the right. Otherwise, everything is same in between D and L-glucose. So, if L-glucose tries to bind with that place, it won't be able to fit. Only D-glucose will be able to fit. Let's suppose there is a disease in a person where that person cannot synthesize proper D-glucose molecules. So those D-glucose molecules will not be able to bind there, as a result of which the function of that binding will not take place. That is, let's suppose after binding over here, this receptor gets activated and this portion of the receptor gets activated, and this activates some other molecule which activates some other molecule, and thus a signal is reached to the nucleus, and let's suppose DNA replication takes place because of that signaling. So because of this binding, DNA replication takes place. Suppose there is a disease in a person where D-glucose is not synthesized properly. So we will supply D-glucose in the form of medicine. When we supply the D-glucose in the form of medicine, we must be very much particular with the three-dimensional arrangement of the constituent atoms. We will only supply D-glucose. If we supply L-glucose, then that problem will not be solved. So the three-dimensional arrangement of atoms is very much important, and that is called as stereochemistry. That is, arrangement of the molecules constituent atoms in three-dimensional space is called as stereochemistry, or it is also called as configuration. So a biomolecule's stereochemistry or configuration is very important because the biomolecules, they interact with each other. So the fit between them must be stereochemically correct. For example, in case of enzyme substrate complex formation, an enzyme binds with the substrate. Let's suppose this is one enzyme molecule and this is substrate, and this is the binding site of the enzyme molecule. The substrate will come and will bind with the binding site of the enzyme. So this binding, this fit must be stereochemically correct. Otherwise, this substrate will not fit with this enzyme. So we must be very much particular with the three-dimensional arrangement of the constituent atoms. For example, in case of enzyme substrate complex, in case of hormone receptor complex, this is of very much importance. In order to study stereochemistry, we should start with isomerism. So we will see what is isomerism, what are the types of isomerism, and when we will study the types of isomerism, then we will come to know how biomolecules are stereochemically different. That is, how their constituent atoms are different and what are the names which we give to them. That is, D-glucose and L-glucose. These are the stereoisomers. So what is the meaning of stereoisomer? So if you will watch this video, then you will come to know the answer of all these things. Isomers are the compounds which have same molecular formula but different arrangement of constituent atoms in space. To understand this, let's take one example. You should look at your hands like this. The molecular formula of your both the hands is same. That is, both the hands are made up of 5 fingers. But when you look at your hands in this way, then the first finger over here is index finger. The first finger is index finger when I look at my hands like this. But of this hand, the first finger is little finger. So the arrangement of the constituent atoms of both these compounds are different. So these both are isomers. Let's understand this with one more example. Let's suppose these are the two compounds which are isomers. How can we say they are isomers? They have same molecular formula. The first one, that is, the carbon atoms in both these compounds are 4 in number. 1, 2, 3, 4. 1, 2, 3 and 4. Hydrogen atoms are 10 in number. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Over here also there are 10 hydrogen atoms. So the molecular formula of these two compounds are C4H10. So if the molecular formula is same, then it is our they are isomers. The second thing, which type of isomer? Over here, the connectivity of the atoms is different. See, over here the straight chain is made up of carbon atoms and the carbon atoms are arranged one by one in a straight line. But over here in a straight line there are only 3 atoms of carbon and one carbon is acting as a substituent to the second carbon. So the arrangement of the atoms is different in both these things, but they have the same molecular formula. So these type of compounds are called as isomers. Now isomers are of two main types, that is structural or constitutional isomer and stereoisomer. Structural or constitutional isomers are the two compounds which have the same molecular formula, the first thing. The second thing, they have different arrangement of constituent atoms. But what kind of difference do they have? The type of difference gives us two types of isomers. That is how they are different, how the constituent atoms are different in three-dimensional space. On the basis of that, we have two types. In case of structural isomer, the constituent atoms have different connectivity. That is, the main chain is of different types. The main chain has different pattern. Over here, the main chain is having 4 carbon atoms, but over here, the main chain is having 3 carbon atoms. So the different arrangement of constituent atoms in case of structural isomer talks about the different sequence or different connectivity in both these things. So structural isomers are the compounds which have same molecular formula but different arrangement of constituent atoms in space. That is, they have different connectivity or atoms are bonded in different sequence. So this is our structural or constitutional isomer. Now stereoisomers. This is our main topic. Stereoisomers. Stereo means space and isomer, you all know the meaning, same molecular formula but different arrangement of constituent atoms. In case of structural isomers, they have different connectivity of the main chain. But in case of stereoisomers, they have same molecular formula, same connectivity. That is, suppose 2 molecules are there and these 2 are stereoisomers. So they both will have the same arrangement or same pattern of carbon atoms in the main chain. But the side atoms would be different. In case of D and L glucose, let's suppose D and L glucose is an example of stereoisomer. So the side atoms are different. They have the same main connectivity but different configuration. That is, the constituents are arranged in different ways in three-dimensional space. Now, stereoisomers are of two types: optical isomers and geometrical isomers. Optical isomers are of two types: enantiomers and diastereoisomers. We will talk about stereoisomers in detail. Now, let's talk about stereoisomers. Stereoisomers have same molecular formula, atoms bonded in same sequence, but the configuration is different. That is, subsequent side atoms are arranged in a different way. Normally, carbon-containing compounds, they exist as stereoisomers, and stereoisomers are of two types: optical isomers and geometrical isomers. Geometrical isomers have these three things, that is, same molecular formula, atoms bonded in same sequence, but different configuration. Apart from these three properties, there is one more property, that is, geometrical isomers also have either a double bond in their structure or a ring in their structure. For example, over here, see, this carbon is double bonded with this carbon is double bonded with this carbon. And these two structures, that is malic acid and fumaric acid, these two compounds are geometrical isomers. That is, they have same molecular formula. See, you can see the same molecular formula is there: 2H, 2COH and 2 carbon atoms. Then they have same atom bonded in same sequence. That is, carbon atoms are bonded in same sequence. The main chain is composed of C double bond C, C double bond C. But they have different configuration. That is, the side atoms H and COOH are arranged differently. Over here, it is this carbon double bond carbon is having H. Both the H are on the upper side and COOH is on the lower side. But over here, on the upper side, this carbon is having H and this carbon is having COOH. Similarly, on the lower side, this carbon is having COOH and this carbon is having H. So this is cis malic acid and this is trans malic acid. So in case of geometrical isomers, there is a double bond or a ring, and they show mainly cis-trans isomerism. And in case of geometrical isomers, the rotation around carbon atoms, that is this C double bond C, it cannot easily rotate. The rotation is very much restricted. So such compounds, they show geometrical isomerism. Optical isomerism is the second type of stereoisomerism. In case of optical isomers, along with these three things, there are two more things. That is, they have chiral carbon atoms in their structure and they can rotate plane polarized light. I have talked about plane polarized light and the rotation, how plane polarized light is produced in my previous video. You can go and check out. So, optical isomers are the compounds which have chiral center in them. That is, the carbon atom is attached to 4 different groups in their structure and they can rotate the plane polarized light either towards right or towards left. So, the optical isomers have 2 more things along with these 3 things. That is, same molecular formula, atoms bonded in same sequence but different configuration. And they have chiral center and they can rotate plane polarized light. Whereas geometrical isomers, they have either carbon-carbon double bond or they have double bond and a ring structure. And basically, carbon-containing compounds exist in form of stereoisomers. So, optical isomers have chiral carbon atoms as well as they can rotate plane of polarized light. Optical isomers are of 2 types, enantiomers and diastereoisomers. In order to understand enantiomer, let's take the example of our hands. These 2 hands, they are mirror images of each other. You just look at your hands and you just imagine there is a mirror in between these 2 hands. And when there is a mirror, you will see the reflection. That is, little finger is over here. Similarly, your little finger is over here also. So, if there was a mirror in between, then that mirror would reflect this hand. So, it would reflect this hand. That means this little finger would be the first finger, then ring finger, then ring finger, middle finger, index finger. So, both these hands are mirror images of each other. But when you try to superimpose them, when you try to put one hand over the other, then they are non-superimposable. You cannot superimpose, that is, you cannot put one hand over the other and allow these hands to exactly fit. These both the hands, they will not fit exactly with each other because these are non-superimposable. So, this is one example of enantiomer. So, enantiomers are the compounds which are mirror image of each other and they are non-superimposable. Because of this only, we need different gloves. We need two gloves for both these hands. If our hands would have been superimposable, then we would have needed only one glove, and that glove we can wear in this hand also or in this hand also. But for both the hands, we have different gloves, a right hand glove and a left hand glove, because our hands are superimposable. So enantiomers are the compounds which are optical isomers, but they are mirror images of each other as well as they are non-superimposable. Diastereomers are the compounds which are non-mirror images and non-superimposable. These are mirror image, non-super. These are non-mirror, non-super. Let's see the one example. Let's take one example of 2,3-disubstituted butane. This is a butane molecule and there are substitution at two places, the second and third carbon, and these substitutions can be anything. Any two other atoms can be substituted on these two places. So we have used X and Y as substitute to represent substituted atoms over here. These are the four types of 2,3-disubstituted butane. See over here, X and Y is substituted in this way. Over here, X and Y is on right side. Over here, X is over here, Y is over here. But over here, X is over there and Y is over there. So there are four different patterns of 2,3-disubstituted butane. Let's search which are the compounds which are enantiomers and which are diastereomers. Let's see. See these two compounds, that is these two forms of 2,3-disubstituted butane are enantiomers because these are mirror images. HH. So HH is on the same side. Then XY, and they are non-superimposable. So if I will try to put this molecule over this, then X will be substituted. X will come over H and Y will come over this second H. This H will be above X and this H will be above Y. So these two molecules are non-superimposable, but they are mirror images. So these two are the enantiomers of 2,3-disubstituted butane. These two are also enantiomers. You can see these two are mirror images and they are non-superimposable. So these are the four different forms of 2,3-disubstituted butane, and out of these four forms, two are enantiomers of each other. See over here, these two are the diastereomers. These are not mirror images. X, Y, H, then H, Y. So these two are non-mirror images and these are non-superimposable. If I try to put this above this, then H will come above X, Y will come above Y, and X will come above H will come above H. So these two are non-superimposable and non-mirror images. Similarly, this one and this one, they are also non-superimposable and non-mirror images. Similarly, this one and this one. So two pairs of stereoisomers, that is this one and this one are enantiomers, and others are diastereomers of each other, that is they are non-superimposable and non-mirror images. If any compound is having chiral carbon atom, then by using one formula we can predict what are the number of stereoisomers of that compound present in nature. That is 2 to the power n is equal to number of stereoisomers. N is number of chiral carbon atom. Let's put this formula and see how many stereoisomers of 2,3-disubstituted butane is possible. Over here, there are only two chiral centers. This is our first chiral center. This carbon is attached with X, H, CH3 and this group. And this carbon is attached with H, Y, CH3 and this group. Whereas this carbon is attached with 3H and this group. And this carbon is attached with 3H and this group. So this carbon and this carbon are achiral because they have similar substituents, that is similar atoms are attached, H is attached. Whereas carbon number 2 and 3, they are chiral carbon atoms because they have 4 different substituents attached to them. So our 2,3-disubstituted butane has 2 chiral carbon atoms. So by using this formula, we can write 2 to the power 2. So we get answer 4. So there are 4 numbers of stereoisomers. Out of these 4 numbers, there are 2 pairs of enantiomers. These 2 pairs, this is the first pair and this is the second pair. So out of these 4 stereoisomers, there are 2 pairs of enantiomers. And all these are diastereomers of each other. That is, this one is diastereomer of this one is diastereomer of this one is diastereomer of this one. So all these ones are diastereomers of each other. So, by using this formula, we can predict the number of stereoisomers of any chiral carbon containing compound. With this, we come to the end of this video. If you want to join my Telegram channel, the link is given in the description box. You can ask me any doubt in that channel. Thank you for watching.